marine lab, after which she completed a PhD at Scripps Institution of Oceanography in 2014. And after that, she pursued a postdoc at Scripps Institute. And now she is an assistant professor at J. Craig Venter Institute. But if I understood it right, you will be going back to Moss Landing, right?
Yep.
Okay, yeah. And Sarah's research focuses on the evolution of marine phytoplankton, the metabolism of and physiology of diatoms, and regulation of eukaryotic gene expressions. And today you will talk about the metabolic insights from nutrient limitation functional genomics studies of model diatoms. So welcome.
Thank you. Okay, so yeah, so the title's slightly different, but it's the same general concept that my interest and area of expertise at this point is really in the fundamental metabolism of carbon and assimilation of nitrogen and metabolism of nitrogen in marine diatoms. But I'm by no means exclusively interested in diatoms. They're just at the moment the most well-developed phytoplankton model to start to use some of these really advanced next level multi-omics genome scale approaches to understand their biology at an unprecedented scale. So that's what I will talk about today. And as you mentioned, I am still in San Diego, in La Jolla. I spent a lot of time at Scripps and I'm now at the JCVI, which is just up the hill, a very short walk. And next fall, I'm going to be moving back to the Monterey Bay area, where I'm going to start my own lab at Moss Landing Marine Labs in fall of 2021. And that's a master's program, but I will also have postdoc positions available at some point, hopefully. So if any of you are interested in coming and spending some time in that world and would like to do a postdoc with me or interact with me academically as a collaborator, I would love that. So please keep me in mind. We're on the edge of the Pacific, which, as you probably have seen by now, is a very large body of water on our globe that has really interesting patterns of chlorophyll, indicative of different phytoplankton population dynamics that are responsive largely to nutrient inputs driven by a lot of seasonal patterns. And so I'm showing this animation to you, which is sea surface chlorophyll or sea color integrated over time, because these patches of phytoplankton are often dominated by diatoms. And diatoms are easier to study than other phytoplankton taxa because of their large cell size. They're often very easily identifiable by their morphology, and they are at the foundation of a really short food web up to fisheries. And so in oceanography, they've been kind of on the radar for a very long time. And so there's a lot of knowledge about their ability to use nitrogen and outcompete other phytoplankton taxa. They're kind of classically known to dominate upwelling systems or after the spring bloom. They're the first phytoplankton often to bloom in HNLC regions, which are regions of the ocean that have high nitrate, low chlorophyll, that are iron limited. So when you add iron to these zones, they're the first phytoplankton taxa that respond. They have these mechanisms for luxury uptake. They can store excess nitrate in their vacuole, kind of like a vacuole that plants have, which allows them to suck up more nitrate than they're using at the moment, outcompeting other taxa. They also can utilize a variety of nitrogen sources, inorganic and organic. They use nitrate, nitrite, ammonium, urea, a variety of amino acids. Different taxa have different abilities to do that. And so what I'm showing here on the right is a micrograph of one of our model diatoms, Phaeodactylum, which I know you probably have heard a bit about by now after hearing from Chris Bowler and Mac and Aaron. So the wild type cell up here is the chlorophyll shown in red, and these green droplets are lipid droplets. This is after the cell has been nutrient stressed for several days. And the bottom cell here is where we've knocked out the nitrate reductase gene. So they're no longer able to assimilate nitrate that they take up. They can take it up from the environment, store it in their vacuole, but they can't take it out of the vacuole to use it for growth. And so what happens is you get cells that swell up, their vacuole gets really swollen, and they kind of look like bowling balls in the later stages, or bowling pins. And so that's an example of the luxury uptake at play. And with the advent of genome enabled investigation, we're trying to figure out what are the molecular mechanisms that explain this behavior of diatoms in nature. And so there are a variety of hypotheses that have been generated, that they have superior uptake or assimilation abilities, that they're especially rapidly responsive. They can activate the genes that are needed to assimilate nitrogen more quickly than other taxa. There may be a seed population of diatoms that they have exceptional mechanisms to survive long periods of low nitrogen. Perhaps they have superior mechanisms to recycle intracellular nitrogen. And so these are all kind of hypotheses that we can test by looking at the genome level. And there are several genomes now of diatoms that are available. The top left is Thalassiocyra pseudonana, the first genome that was sequenced in 2004. And then the one just to the right of that, let's see if I've got I can turn on my laser pointer. There we go. Is phytodactylum. And I would say that right now, despite being the second diatom that's sequenced, is probably the most well-developed model, because there are it's really easy to grow. There are a ton of molecular tools that have been developed to engineer the genome. And it's kind of a little bit easier, and there's more literature on phytodactylum at this point. There's Thalassiocyra oceanica. All of these diatoms have been selected for different reasons. Thalassiocyra oceanica does really well in low iron environments, and so its genome was sequenced to specifically look for mechanisms it might have that would explain that ecology. And there are other, this is a diatom from Lake Baikal. These two down here, Fistulifera solaris and Cyclotella cryptica, are both really exceptional diatoms at accumulating oil bodies. You can actually see them in these images. So the part where they don't have the brown, which is the photosynthetic pigment, are actually big droplets of oil. And this oil is chemically very similar to the precursor for fuel that we can use as a stock to make renewable biofuel out of. And so these organisms have been sequenced particularly to begin to explore their metabolism and perhaps optimize their metabolism through engineering for the development of renewable biofuels. And then there are other organisms that are also of ecological significance. This is Fragilariopsis that does really well at the poles, et cetera. So we have a really rich set of diatom genomes now, with more and more coming online kind of every day. This is probably already even outdated by now. So it's a really rich resource to exploit. And some of the early insights from diatom genomes were reflective of their unique evolutionary history. And so this is kind of a point that I think is really, really important to make. Diatoms, even though they're photosynthetic, are very, very different metabolically than plants. And part of the reason for that is that plants are derived from the primary endosymbiosis, whereby a heterotrophic eukaryote that was not able to do photosynthesis engulfed a cyanobacterium, enslaved it as its organelle, transferred genomic material to the host genome, and that went on to become the progenitor plant cell for the Archaeplastida, or the early lineages, the red algal lineage, the green algal lineage, et cetera. Diatoms arose through a secondary endosymbiosis, where a secondary heterotroph, shown here in this graph, engulfed a fully developed red algal endosymbiont, transferred genomic material to its host nucleus. And so the extant diatom organism, the nuclear genome encodes a variety of genes that are both plant-like from the original progenitor plant cell, and potentially more animal or fungal-like from the heterotrophic host. And to complicate things further, there seems to be some kind of signature of a transient green endosymbiont. And so genomics can really help us address the origins of diatoms and where have they gotten a lot of these genes from. They also acquired a whole bunch of bacterial genes along the way. So they're, a real chimera, a real smorgasbord of functional traits. And another thing that's important to note is that they have kind of complicated intracellular compartmentation that results from this unique evolutionary history. They have extra membranes around their chloroplasts. And in order to target the proteins to the proper place, so even though they have chloroplast genes that are encoded in their nuclear genome, often those are genes that are important to have the protein functioning in the right organelle. And so they've developed mechanisms to target those proteins after they're made back to the original organelle. And that's where it is depicted here with these transit peptides that target them to their appropriate locations. So they're really complicated, and they're a combination of traits. They're not plants and they're not animals. And one of the most interesting findings or surprising findings from the initial sequencing of the Thalassiocyra genome in 2004 was the discovery that diatoms have an animal-like urea cycle. And if you remember from kind of human biology, a urea cycle typically functions to allow organisms to excrete excess nitrogen. And that's a little bit surprising to have seen in a diatom where the populations are often nitrogen limited. And so the question became, what are they using it for? They're probably not just excreting nitrogen out into the seawater willy-nilly. They're more likely trying to hold on to it. And so further investigation led to my supervisor at JCVI and my postdoc advisor, Andy Allen, to make this conclusion that the urea cycle serves as a distribution and repackaging hub for inorganic carbon and nitrogen. And it basically allows them to respond quickly to episodic inputs of nitrogen or survive long periods of nitrogen deprivation. So we got some insights into its function there, but I think Andy would be the first to admit that we still don't know a ton about the urea cycle. Other kind of interesting features of diatom metabolism have been summed up really nicely in this 2017 theme issue, which I love the name. It's the peculiar carbon metabolism in diatoms. They do a very odd configuration of carbon metabolism with pathways in places that they're not supposed to be, likely as an artifact of all the rewiring of metabolism that happened as they evolved. One of the really interesting features is that their glycolysis pathway, which is, if you've taken metabolic biochem, is a central pathway that almost all organisms have to allow for catabolism of sugars and sustaining of energy. They have the lower half of glycolysis in their mitochondria, which is a very, very unusual thing in eukaryotic evolution. So, what's the advantage of that? We don't know. So metabolism overall is super difficult to give a talk on because it's so detailed and so complicated and not in and of itself extremely exciting, unless you're a big metabolism dork like I am. But I thought this graphic, which summarizes general cellular metabolism, so this is not specific for diatoms, this I think is modified. It's kind of a amalgamation for a lot of different organisms, but it depicts metabolism or cellular metabolism like a metro map. And I really like this because you can start to layer on all the levels of regulation and expression and make some analogies as to, what's actually happening at the level of metabolic flux. So if you think about like people moving from one place to another as metabolic flux, you might need to accomplish everybody moving from their house in the morning to their workplace in the day and then home in the evening. That's what a cell has to do as it goes through a diel cycle. It has to orchestrate all of these events so that things get to where they need to be and that the cell functions as a whole. And if you think about that analogy and kind of take it a little bit further, you've got the genome, which would represent the actual physical stations and tracks of the metro. You've got a transcriptome, which is the train schedule, the way that the trains are supposed to run. You've got a proteome, which is actually where the trains travel from one station to another. A metabolome would be kind of the amount of people in the station. So you can imagine this like pyruvate station, for example. There might not be at any given time a ton of people in the station, but it's a really busy station, so there are a ton of people moving through it constantly. So the flux through that station might be really high. And pathway usage is the movement of people. So, there's the red line. A ton of people are moving through the red line. It's a really busy line. And so the multi-omics approach, when you make this analogy and kind of take it back to the actual cellular level, is trying to understand in a diatom cell what's happening at the genome level, the transcriptome level, the proteome, the metabolome, and the pathway usage level, and integrating all of that information. And when you do it on a map that's really complicated like this, it becomes kind of a bigger thing than any human brain can really contain. And so we tend to defer and rely on systems biology developed approaches where computer models can take all of this information into account and model it for us. And so that's kind of what's getting me to this idea of a genome scale model, which is a tool that we've used or begun to use in diatom biology to better understand the pathway usage in these organisms under shifting environmental conditions. For example, the shift between nitrogen deplete or replete, or silicon deplete or replete, et cetera, or excess light, to understand why they have these unusual pathways and these unusual configurations. But it's also a really valuable tool in engineering metabolism. So if you're, for example, trying to optimize the production of fatty acids or triacylglycerols for lipid production, you might go in and try and say, okay, I'm going to overexpress fatty acid biosynthesis at the transcriptome level. And you could put that into a genome scale model and test that hypothesis and see if the whole metabolism, the whole network of the cell actually supports flux to that pathway through that kind of engineering approach. So it's a really effective tool for targeted metabolic engineering. But what is genome scale model? I'll just run through this briefly. This is a really nice graphic from Manish Kumar, who's preparing it as a chapter for the Molecular Life of Diatoms book that will be coming out probably next year. And there's a variety of steps that need to go into curating these models. They were initially made for bacteria, and bacteria are relatively simple. They don't have the compartmentalization oftentimes that diatoms do, or these kind of very discrete organelles. But in order to do this, a genome scale model, you need a genome sequence. You have to assemble the genome and annotate it. All that functional annotation gets fed into a metabolic model where that you construct the network, and this is all in silico. And then using reaction information from different databases, you generate a matrix of these reactions, constrain the model with actual data. How much nutrients did you give the organism? Have you collected any other kind of ancillary information about metabolite pools or transcript abundance, et cetera, that you can use to constrain all of the areas that equation can be solved? And then you solve this equation in a solution space that then gives you like a flux topology. And so this graphic down here is the subcellular localization of the mitochondrial glycolysis pathway. And that was one of the pathways that early on in the construction of these genome scale models, the systems biology people, I think, were a little surprised to put that pathway in that organelle, since that hadn't been seen before. But that sort of thing is really important because that lower half generates a lot of energy. And if you put all that energy generation in the cytosol versus the mitochondria, you're going to get a really different topology of flux within the cell. And so this is a graphic that's been kind of central metabolism for phaeodactylum, compartmentalized. You've got mitochondrial glycolysis here. This is not depicting the urea cycle, Calvin cycle in the chloroplast, et cetera. And so you do construct the model, do your experiment, constrain the model, and then you get, at the end of the day, these flux predictions. So each one of these is the reaction, and then the protein ID that's associated with that reaction. And then it can give you flux predictions of flux through that reaction under a variety of conditions. So here we're comparing nitrate and urea. So I know I'm kind of using up a lot of my time real fast, but I'm just going to go through a very quick overview of some real world insights that we got from using this approach in phaeodactylum. And this paper was published last fall, so you're welcome to take a look at it, and if you're more curious than what I'm able to present today. But what we did with phytoplankton was we developed this compartmentalized model of nitrogen metabolism. We generated 42 phylogenies of a bunch of nitrogen genes and validated their subcellular localizations and modified the genome scale model accordingly. We did a lot of functional genomics, including transcriptomics and proteomics. We did some metabolomics also. And another key thing that we did was fluxomics. We labeled nitrate and urea substrates and then fed that to the diatom cultures to validate some of the predictions that were made with the genome scale model, since those are just, at the end of the day, simulated fluxes. So this is what this looks like, kind of a smaller version of the Metro map tailored for phytodactylum. There are a few interesting features. Nitrate is assimilated in the chloroplast. Urea is assimilated in the mitochondria. Each organelle has its own GS-GOGAT cycle, which functions to take ammonium and assimilate it into glutamine, two amino acids, the first committed step of amino acid biosynthesis. Each organelle has these ALT, AST kind of systems. There's an ornithine biosynthesis pathway in the chloroplast, and then of course the urea cycle in the mitochondria. The phylogenies told us that some of these genes came from the endosymbiont and some of them are present in the host to begin with. What was really interesting is that the nitrate transporter that diatoms have is more host-like. So it indicates that before they acquired the chloroplast, they were already able to uptake and store nitrate and assimilate nitrogen from nitrate. And that the endosymbiont made arginine in the chloroplast. So this ornithine biosynthesis is the first part of the arginine biosynthesis pathway. But they don't have the second half of that plant-like pathway. And so the diatoms lost that. And it also shows that they have this ability to utilize urea that was acquired with the endosymbiont. So our transcriptome experiment is shown here. This is a pre-treatment, and then we had 4 experimental conditions: no nitrogen, nitrate, nitrite, and ammonium. And we took gene expression and proteomic data from 15 minutes, 45 minutes, and 18 hours of each of those conditions. I'm not going to talk about the details of it, but the main feature here is that nitrogen status, independent of nitrogen source, really is what's driving the overall patterns of gene expression at both the transcript and protein level in phytodactylum. So we use that data to constrain the model, the genome scale model. But we also looked at just the transcript level expression. And so here's nitrate assimilation genes, ornithine biosynthesis, ornithine urea cycle, and urea assimilation genes. And they partition into 2 main patterns: nitrogen replete and deplete, as I mentioned. But one thing that's really interesting to note is that here's the urea cycle. And most of the urea cycle is upregulated when nitrogen is present, except for this final step, arginine. And what you can kind of predict about the flux from this pathway is that the nitrogen assimilation, when they're growing on nitrate, is happening in the chloroplast like normal. They make ornithine, which then might get exported to the mitochondria, where it enters the urea cycle. But it doesn't complete the urea cycle. So this breakdown of arginine back into ornithine, the transcript data doesn't support that actually happening functionally. And we kind of have seen this broken expression of the urea cycle before. This is some work that we did with iron limitation over day-night cycles in phytodactylum. And that whole first part of the pathway is upregulated during the day. And then that last step is upregulated at night. And that's consistent with a flip-flop in the types of metabolites that you would expect to accumulate to be synthesized and be consumed under those conditions. And so what we concluded was that the urea cycle actually is functioning as an arginine biosynthesis pathway. And as I mentioned, they have this first half of the plant-like pathway, and it looks like the second half, which they obtained from the host, is how they actually make ornithine. And so diatoms do it differently than plants, where everything is in the chloroplast. Animals have a whole first half of the pathway that's different, that diatoms don't have, but diatoms do have this second kind of half. And so they make ornithine in the chloroplast, send it to the mitochondria to make arginine, and they integrate this metabolism across these organelles in a really, really interesting way. This is just N15 data to show that we validated some of the flux predictions from the model, which I'm going to show here. So these are the actual flux measurements that were calculated from simulations on nitrate and urea. And there's two kind of main salient features. And this is a lot of information. I wanted to show you that, you have to pick through a lot of cells in Excel before you get down to what the really meat of it is. But you can kind of break that down into the real take home. And one thing that this data is showing us is that nitrate is assimilated in the chloroplast. But in order for that to happen through this GS-GOGAT and aspartate transaminase system, they need oxaloacetate, which the chloroplast doesn't produce. So the mitochondria delivers pyruvate. Pyruvate gets converted to oxaloacetate, which supports the assimilation of nitrate. And then nitrogen is exported to the chloroplast in this aspartate shuttle. When they're growing on urea, it's different. Urea is assimilated in the mitochondrion through a similar system, but then in order for the chloroplast to get nitrogen, the model simulates that it gets transported over as alanine, where the alanine system can recover it as glutamate to support nitrogen assimilation in those compartments. So the details of this are not important, but what I'd like you to kind of focus on from this is that it taught us a lot about how diatoms are exchanging carbon skeletons and amino moieties, or just nitrogen groups, between the organelles in ways that we wouldn't have been able to anticipate necessarily just from transcriptome or proteome alone. And then when we went back and we looked at the transcript, these patterns really made a lot of sense and were very consistent with the flux predictions. And so we feel like, at least in this case, the flux simulations were giving us a really good idea of what was actually happening in the cell. Yeah, okay. So I'll wrap up there. I just want to summarize that diatoms, have really compartmentalized nitrogen metabolism that retains characteristics from both the host and the endosymbiont, which presents a lot of challenges, but is really exciting in construction of these GEMs and FBA. And I know you all have probably heard about cyanobacterial endosymbionts and diatoms, and there are, really exciting opportunities to make these kinds of genome scale models, considering cyanobacterium as another compartment or an organelle, and also considering exchange within organelles, but also to the community. So you can start to make genome scale models that don't only simulate a cell, but can simulate a community, a microbial community. So that's really exciting. These systems biology approaches have revealed, I think, the most interesting take home from this is that metabolism in diatoms is really integrated between the chloroplast and the mitochondria in ways that other organisms that we know today don't do it. They have mitochondrial glycolysis, their urea cycle and arginine biosynthesis is split. They've got these aspartate and alanine shuttles. And overall, this strategy of linking metabolism between these organelles kind of streamlines communication among the cell about the macromolecular status and energy status. So, once the chloroplast is assimilating nitrate, it relies on a coordinated metabolism of the mitochondria to make that happen. And the chloroplast and mitochondria are the major energy generating and consuming organelles in the cell. And so this streamlined metabolism, we hypothesize, may explain why diatoms can outcompete other taxa. And we now have kind of a molecular mechanism to address that. So that's the real big take home, emphasized with a PowerPoint animation. So just to wrap up, I want to quickly acknowledge Andy Allen, who I've done all of this work with at the JCVI, and co-authors on these studies, and also to the Molecular Life of Diatoms community, which I think is a really, really wonderful community of people that I've learned a ton from and have had a lot of fun working with, and who you have heard from a lot by today. So with that, I'll just end, and I don't know if we have time for questions. I think I went over, sorry.